So today we start a conversation about metabolic strategies. If you're familiar with cell respiration, it's basically a recap of what we were talking about before. Some of it like enzyme stuff, things to remember when we get to antibacterial drugs, so that will become easier. Metabolism is the term for all cellular processes in the cell. Okay? And reactions in the cell can be either reactions of degradation, that we call catabolism, or reactions of synthesis, that we call anabolism. Now, catabolic reactions, shown here, are mostly exergonic. So here on the right hand side you can see highlighted in red are catabolic reactions, reactions of the breakdown. For instance, in the glucose catabolism, which results in production of CO2 and water, energy is released. And this energy released in a catabolic reaction is then used by anabolic reaction of ATP synthesis right here. So anabolism is mostly endergonic. If you think about other examples, it can be, for instance, breakdown of lipids by beta oxidation, free fatty acids broken into individual molecules of acetyl-CoA. And this reaction is hugely exergonic. It produces a lot of energy. And this energy can be used to fuel ATP synthesis, which then, in turn, ATP then, can be catabolized to fuel anabolic reactions again, such as polysaccharide synthesis. Thank you. Such as polysaccharide synthesis or protein synthesis, or DNA RNA synthesis. Okay, good? That makes sense? Awesome. Now you have to understand that exergonic and endergonic reactions are coupled reactions. By coupling, I mean that, rarely cells break down stuff just for the sake of breaking down stuff, releasing energy in the environment. So one reaction that releases energy will be coupled to another reaction that uses that energy to produce some complex molecules, right? Does that make sense? It's energy transfer, basically. Energy transfer is not 100% effective ever. And the difference, this is reflected on the picture here. It's really nicely reflected in the left part and absolutely terribly and mistakenly reflected in the right. So don't look at the right, like forget about it, okay? So here you see the large arrow that represents catabolism, breakdown, exergonic reaction. So if you think about the size of this arrow as the amount of energy, it's quite a lot. But then this anabolic exergonic reaction cannot use everything. Does that make sense? So the difference will be lost as heat. You can see something analogous every time when you drive a car. Not all the energy of gas is converted into mechanical energy of wheel rotation. A huge portion of it, actually, like 80%, is converted into the heat. Okay. So ATP synthesis, and we focus on ATP a lot because it's a key molecule and we're going to be talking about how to make it. ATP synthesis is anabolic endergonic. So it's the reaction of synthesis that requires energy. Anabolic synthesis, endergonic, uses energy. But ATP synthesis, that it uses energy to store it. ATP is the storage molecule. It's not the ultimate goal of the cell. And ATP hydrolysis is catabolic exergonic reaction. So it releases energy. That's when you take this energy out of the storage, when you break down the molecule of ATP. And thus energy of ATP breakdown can be used to fuel various cellular reactions like transcription or translation. Does that make sense? So I don't think that anyone here is going to argue that all living organisms, and even non-living like viruses, they need energy. But we're going to be focusing on living ones. But in order to replicate and grow, living organisms not only need energy, that they also need building blocks. They need organic carbon. Like when you build a house, even if you have a generator that will fuel all your tools for building a house, if you don't have concrete, wood, all those building materials, you won't be able to build the house out of the pure energy. Okay? So cells are the same. In order to grow and replicate, they need energy and carbon. There are two possible energy sources in the environment. One is chemical. One is light. So organisms that use chemicals as the energy source, called chemoautotrophs. Organisms that use light as the energy source, called phototrophs. You with me? Now, chemicals that can be used as the energy source may be organic or inorganic. Right? For example, like humans use organic chemicals to fuel carbohydrates, lipids. Okay, there are bacteria that can use the energy stored in reduced forms of different chemicals like hydrogen sulfide to fuel cellular reactions. Okay. Now, what about carbon source? Organic carbon is 4-valent carbon that forms tetrahedral, that has tetrahedral orientation of covalent bonds. No, you don't need to know that. Yes. No, you don't need to know. Don't worry about it. What I'm trying to tell you is that organic carbon is very specific, very peculiar. It's quite unique. Okay? So organic carbon can be acquired directly from other organic sources. And so basically by quote-unquote eating organic matter. In this case, organisms that eat other organic matter called heterotrophs. So they acquire carbon from organic sources. The good examples of heterotrophs, and I try to focus on microorganisms, are fungi. They are by definition heterotrophs. And I want to make sure that we're all on the same page with the term organic carbon. Doesn't only mean, I don't know, dead tree or a corpse of an animal, which is undoubtedly organic, bless you. It means tires, like car tires. That's organic chemical. Rubber is organic. And if you're curious, there are fungi that can eat rubber? Yes, there are. Okay? They not doing it extremely fast, they do it. And there's a saying among mycologists, people who study fungi, if there is an organic source of carbon, there will be a fungus that can use it. They very good at it, right? Second option is to use inorganic source of carbon. Organisms that can use inorganic sources are called autotrophs. Auto means what? Self. Self, by itself, right? Meaning that they can take inorganic carbon and convert it to organic carbon by themselves. Does that make sense to you? That conversion of inorganic carbon to organic carbon is called carbon fixation. In general, in biogeochemistry and ecology, conversion of some inorganic compound in the form that can be used in the cellular metabolism is called fixation. There's carbon fixation, there's nitrogen fixation, there's sulfur fixation. Okay? That makes sense to you? So the most obvious example of autotrophic organisms are plants. But since we are not focusing on plants, cyanobacteria. Cyanobacteria are perhaps the oldest, evolutionary oldest organisms on Earth. And we will have a chance to talk about what the primeval Earth looked like when we get to photosynthesis. It's a pretty interesting talk, really. But you understand, right? So this cyanobacteria, they will use light as the source of energy, so they phototrophs, and they will use carbon dioxide from the air as the source of carbon. Does that make sense to you? So when we combine the terms for the energy source and the carbon source, it becomes photoautotrophs. Some microorganisms, like heliobacteria or different non-sulfur bacteria that do not produce sulfur particles, called photoheterotrophs. Okay? Now, what about chemotrophs? So, in case of chemotrophs, you can have Chemo Heterotrophs, sometimes referred to organotrophs, meaning that they use organic source of carbon and, honestly, most frequently, organic source of energy. So what belongs to Chemo Heterotrophs? All fungi, vast majority of bacteria. Not all of them, absolutely not. Green sulfur, purple sulfur, non-sulfur bacteria, they're not. But there are, the majority of bacteria are Chemo Heterotrophs. Okay? Protozoa, all of them, Chemo Heterotrophs. Does that make sense? In my opinion, one of the most fascinating types of microorganisms are Chemoautotrophs. Here. They use inorganic source of energy, and they use inorganic source of carbon. And they are often called lithotrophs, where prefix, it's not a prefix, it's a compound word. So litho in Greek means stone. So they sometimes referred to as rock-eating bacteria. Don't take it too literal. They're not going to munch through the rocks. But I want to look at this specific type of microorganisms. And from the name, try to figure out what would be the environment in which these microorganisms thrive. So first of all, they're using inorganic chemical source of energy. What's the alternative? What are two alternatives? Possible alternatives.
We could use
organic source of energy, or they use inorganic or light, right? So they use inorganic chemical source. The alternatives are light organic chemicals. Believe me, they oxidize hydrogen sulfide to sulfuric acid, not because it's awesome. They do it because light, inorganic matter, is not abundant where they live. Make sense? For that same reason, they have to fix inorganic carbon to produce elementary building blocks of biological macromolecules. Because organic carbon is really, really scarce in their habitat. What is that terrible place on Earth? There's no light and very few organic molecules. Bottom of the ocean, yeah. You don't have to go that far. But think about this. These places have to have an abundance, not just your regular concentration of inorganic chemicals in the environment. They should be so, enriched with sulfates, carbonates, nitrates, all those minerals that this bacteria can use, right? It turns out those are not really bacteria, those are archaea, mainly. And they will live in the vicinity of hot springs. Not just hot springs, but hot springs, volcanic vents at the bottom of the ocean. We're talking totally different temperatures there. We're talking like 500 degrees Fahrenheit. That's overheated. It's not steam because of the pressure. It's basically water. And it's boiling, but it's different type of boiling. And you know what's interesting? Yeah, those bacteria thrive there. You know what else thrives there? Tiny little shrimps that eat that bacteria. And crabs that eat shrimp. And giant tube worms that eat bacteria as well. So actually, those habitats they're not super diverse, but they're pretty rich in life. Because a lot of chemicals, maybe not the most benign, but still a lot of chemicals, a lot of energy sources, very high temperature, that actually allows those organisms to be metabolically active. Okay? So they're pretty rich. Despite of extreme environments. Okay? What I want you to know. Terminology. Exergonic, endergonic, anabolic, catabolic. Kind of obvious. In terms of this, I believe everybody is sitting here waiting. Do we need to know the examples? No. You don't. Okay? Maybe except like fungi, because that's so obvious. Okay? But I'm not going to ask, purple non-sulfur bacteria what? But what I can do, again, I can give you a description. Detailed description of metabolism. This microorganism, such and such microorganism, uses inorganic chemicals as the source of carbon and light, as the source of energy. Whatever. Tell me what's this. Autotrophs. Does that make sense? So match the term to description. Clear? Which brings us to the conversation about oxidation and reduction. That's worse than when my 5th grader had sexual education at school. That was less hard to proceed through. Okay, so oxidation and reduction. I want you to know the terms oxidant, reductant, oxidation, reduction. Okay, so look at the panel here on the left. So this is your oxidant. Okay. Oxidant gains the electron in the process of reduction. And when oxidant gains the electron, it is, okay, wait a minute. So oxidant in the process of reduction is reduced. Does that make sense to you? Now reductant in the process of oxidation loses the electron and reductant is oxidized. That makes sense. Now, I always say that terms in oxidation and reduction, it's not a biology or chemistry problem, it's an English problem. So let's say I'm paying for service I'm paying to Brian. Okay? So I pay him, he gets paid. Does that make sense? From standpoint of conventional English. Right? So say I'm a reductant. Brian is oxidant. I give electron to him. So Brian receives the electron. He's an oxidant for that matter. Okay? So he oxidizes me. And I get oxidized. By giving Brian an electron, I reduce him. And since I reduce him, he gets reduced by me. Do you see that language component here? The active voice versus passive voice. I do something and somebody else does things to me. Okay? You understand what I'm trying to say? Why I pay so much attention to this? Because I'm going to ask you questions in terms of what happens to such and such molecule in such and such process. And you will have to tell me it's reduced, it's oxidized. Okay? So basically, when molecule is receiving an electron, it is reduced. When molecule loses the electron, it is oxidized. So far, does that make sense? And I don't want to make it all rainbows and unicorns, but it's more of a language concept rather than chemistry concept, and you have to get comfortable with it. And we're gonna do a quiz on it, and don't hesitate to ask me, like, do I get it right? And I'm gonna ask you, is this molecule reduced? Is this molecule oxidized? Okay, so we can have some practice. Now, one really important feature of oxidation and reduction, that these two reactions, they are coupled. You can't get electrons from the thin air. Is that understood? Electrons aren't just floating around free. So if one molecule gains the electron, what happens to some other molecule? Loses it. So this is why we call these reactions often redox reactions. So one molecule loses electrons and another one gains them. So reductant loses electrons and oxidant gains them. We're clear? Why we pay so much attention to seemingly fundamental and vague concept of electron transfer between the molecules? There are two types of people. There are always two types of people. Biologists and chemists, in terms of how they approach cellular respiration in the cell. Biologists will tell you that cellular respiration is all about transfer of hydrogens in the cell. Chemists will laugh maniacally over that statement and will despise this statement because it is chemically wrong. Energy transfer in the cell, cellular respiration, is all about electron transfer. Please don't tell Dr. Landis that I said this. I'm physically inferior to him, and I don't want him to beat me to death because he's a biologist. I'm a chemist in this approach. So There are reasons. So biologists focus on hydrogen transfer because hydrogens means acid, acid means death. Okay, so we need to control hydrogens. For chemists, the death is represented by electrons. Because free electron, it's, I don't know, like a 5-year-old after the sugary drink in the store that sells crystal. Okay? It's going to be the same effect. Everything's going to get broken in the cell, right? This electron will bounce off everything, destroying all kinds of molecules, right? So, as a chemist, I need to control my electrons. And as a chemist, I'm telling you that electrons in the cell are being transferred from reductant to oxidant. And as they transfer, they continuously lose energy. Think about this as reductant having high energy electrons and oxidant having them at low energy level. That makes sense. So another analogy that I want to throw your way, imagine that reductant is on top of the building and oxidant is at the bottom. So reductant throws basketball and oxidant catches it. As basketball flies from top to bottom, it gains an enormous amount of kinetic energy. If oxidant that catches the ball at the bottom will not be able to dissipate it, basketball will kill it. Okay? Makes sense? So the idea of energy transfer in the cell is that you have high energy level electrons, and they get transferred to low energy level. And during this transfer, energy will be released, and this energy can then be used for the ATP synthesis. Does that make sense? So it's all about electron transfer. Kind of glimpse into the future in our conversations, majority of our conversations. The molecule that will provide all those high energy electrons will be glucose. And the molecule that will receive all those electrons at the low level will be oxygen. Now, last concept for today. As I mentioned before, electrons are like wrecking balls if they travel by themselves. They can't actually. They won't travel far. In order to carry electrons, almost physically carry electrons from one point to another in the cell, and those are physical distances, they're really, really tiny. We're talking about nanometers, but they still exist. In order to carry electrons from one reaction to another, from one molecule to another, from one protein complex to another, you need to have some kind of electron carriers. There are three, well, I mentioned only two here, there are 3 electron carriers that we will mention in this chapter. It's NAD+, FADH, and another one that we will find later in photosynthesis is NADPH. My smiles on some of your faces, I see that. This sounds unpleasantly familiar. So, on the left, a formula of NAD+. Well, not the entire NAD+. Okay? It's just the nicotine molecule, because NAD stands for nicotine adenine dinucleotide. No, you don't need to know what it stands for. No, you don't need to know the formula. But you need to know 3-letter acronym, NAD+. NAD+ receives 2 electrons, and the hydrogen, it actually should receive 2, but it's a bit of a mistake here. Okay, and it becomes NADH. Now let me ask you this. When NAD receives electrons, and refer to the left panel as well. When NAD receives electrons, NAD+, is it reductant or oxidant? NAD+. NAD plus receives electrons. Look at the left panel. What receives electrons? What receives electrons? The oxidant. Okay? But the process in which it receives electrons is called what?
Reduction.
Reduction. And it means that oxidant, or NAD plus, is being what? Reduced. In the process of reduction, NAD plus, an oxidant, is being reduced. Therefore, we call this form, or this form, a reduced form. In the opposite process, when NADH releases electrons, NADH will become a, it's always reduction oxidation. It will become what?
An oxidant. Or
NADH.
reductant.
NADH is reductant, and it will be oxidized to its oxidized form, which is NAD+. If we look at FAD, another electron carrier, FAD is oxidant that receives 2 electrons. It becomes reduced and forms FADH2, its reduced form. When the reduced form, FADH2, loses 2 electrons in the opposite reaction, it becomes FAD, the oxidized form. Throughout the discussion, the conversation about cellular respiration, we will mention these electron carriers many, many times. And every time I will be asking you, is that a reduced form? Is that an oxidized form? In this reaction, is NAD reduced? Is NADH oxidized? Or what happens to those molecules? So we can practice, so when time comes, you know all of it. Does that make sense? Have a good weekend. See you on Tuesday.